Yes, ma'am. Just a minute.
Sabiha, you also? No. Okay, students. So we had discussed about photorespiration and how it is a very wasteful process which decreases the productivity of the plants. Today, our topic will be the factors affecting the photosynthesis. What are those certain things that can affect the rate of photosynthesis? Meaning, it can either increase how much photosynthesis is done or it can decrease how much photosynthesis is done. So, there are all in all these four factors that affect the rate of photosynthesis. We have light, we have carbon dioxide concentration, we have temperature and water. Now talking about the light. So light, as the light intensity, meaning the intensity or the strength or the amount of light. So whenever the light intensity increases, the rate of photosynthesis also increases. Okay, simultaneously both of it will increase, but only up till a certain point. Meaning, once the light saturation, meaning the amount of light that is required by the plants, once that is reached, then no matter however much light intensity will increase, the rate of photosynthesis will not increase in that way. And you people might remember that there is like a term called as photooxidation of the pigment. What is the meaning of that, students? What is the meaning of?
Chlorophyll A, ma'am.
Yes.
Chlorophyll A.
Yeah, that's true. Chlorophyll A. Photooxidation means sort of like destroying the structure of the pigment of chlorophyll A. Okay. And all of you just disappeared. Come on, students, switch on your videos. Quick. I think I've told it like I've asked you to do so at least 5 times. Switch it on. Yes, Jobita, you also? So this the first case was of light, because if the light intensity will be increased a lot, then what will happen? Ultimately, the photo oxidation of the pigment will also happen. And remember that it doesn't mean that if we increase the light intensity a lot, so the rate of photosynthesis will also increase a lot. No, it only increases up until it reaches a certain point. That is the light saturation. You people understand saturation? Do you remember, like, when I teach a lot of the things to you, then I ask, like, have you people reached the saturation point? You remember that? What is the meaning of it? What is the meaning of reaching a saturation point? Tell me.
It's like reaching your limit. Like you can't handle it anymore.
Exactly. Does that happen to you all, students? Saturation
Mm.
point? How many times do you reach your saturation point in a day during the tuition? Hmm? Okay. Okay, it varies from person to person. A person can reach a lot. Even I reached my saturation point. It's not just for studying. I reach my saturation point for teaching also. And I'm like, I'm done. My throat is paining and all of that. I reach my saturation point while I have to clean my room. That is also sometimes I reach it. So it varies. But reaching a saturation point means now no more you can do that particular task. Same way, when the light cannot be absorbed more than what is needed, then that is a light saturation point, meaning now the plant does not require more light. So the first thing is that if you increase the light intensity, the rate of photosynthesis will increase, but it will only increase up until it has reached the light saturation point. First point is clear, light, how light is affecting photosynthesis. All right. Now let us see how carbon dioxide affects the rate of photosynthesis. Now see, what happens is that when we will increase the concentration of the carbon dioxide in the atmosphere, the rate of the photosynthesis, just like light, will also increase till the compensation point. Again, there is a saturation point for CO2 also, meaning the plant will not require more CO2 than what it has already taken in. So that is CO2 concentration. Then comes the temperature. Now, temperature, it does not influence the rate of photosynthesis directly, meaning even if it is cold, even if it is hot, even if it is winter, this thing, autumn, spring, any season you say, photosynthesis keeps on going, right? Photosynthesis happens. Now, the direct effect of temperature is not there for photosynthesis, but what happens that you people, do you remember when we were talking about the proteins, what happens to proteins when you increase the temperature higher than the optimum temperature?
They get denatured.
Yes, they get denatured. And tell me what will happen if lower temperature than their optimum temperature is there?
They're inactive.
They get inactive, right? Both the cases or both the situations are detrimental, meaning it's of like it's bad for the enzyme. So when we talk about the temperature will not directly affect photosynthesis, but indirectly it will affect, meaning if high temperature is there, the proteins, the enzymes will not work efficiently. If low temperature, very low temperature is there, then also the protein will not work efficiently. So all in all, we can say that temperature has an indirect effect on the photosynthesis, that mostly in the dark reaction, because we know that the enzymatic activities, all of it takes place in the dark reaction, isn't it? What is the other name for dark reaction, students? Tell me.
Light independent, ma'am.
Light independent. Okay, just like how light reaction is called as photochemical stage. What is the name of dark reaction? It is biochemical. Okay, biochemical. Why? Because, and tell me one thing, students, if the name of the reaction is the dark reaction, does that occur in the dark?
No, ma'am.
Then why is it named as dark reaction?
Because it can't take place in the dark.
That's why its name is dark reaction. See, what happens
Because
is that, tell me,
because
say.
it like does not depend upon light products.
Exactly. Because it does not depend on the light directly, isn't it? But it depends on the products of the light. What are the products of the light, students? Products of the light.
NAD.
ATP and NADPH. Right? Two products are there. And using those products, the you can say the biochemical phase or the dark reaction takes place. Now, the next factor that affects the rate of photosynthesis is water. Now, due to the increased amount of water, the rate of the photosynthesis does not increase proportionally, as after saturation, no more water is required. Same thing, just like how light was there was a saturation point. Same way for water also. Now, you might have seen that if you water the plant much more than it is required, do you see that it flows out of this? It starts flowing out of the soil or pot. It starts leaking, right?
Yes.
So that is happening because why? Because the plant doesn't need much more than that, right? So like the plant will utilize as much as it needs, but not even a tiny bit more than that. Now, after that comes the Blackman's law of limiting factor. Now, this law tells us how one of the factor can be a limiting factor. See, so this law states that if any chemical process is affected by more than one factor, just like in the case of photosynthesis, it is affected by light, temperature, CO2 concentration, right? All of these things, water. So if the chemical reaction is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. Nearest to its minimal value, meaning if direct, you can say, change in the factor will change the rate of photosynthesis, then that becomes the limiting factor. Meaning because of it, the changes will take place. And if it is not there, the change will be normal. So that is the law of limiting factors, and it was given by Blackman. So it is famously called as the Blackman's law of limiting factors. Clear, students? Any doubts? So with this, we have come to the end of the chapter also. Now you people have to note it down. Apart from that, you people tell me which chapter should we start next.
What, ma'am?
Which chapter shall I start next with you all? Is there any chapter preference or should I choose my favorite? See, look at your the list of the content that you people have in your book. See and tell me. Also, students, have your half yearly exams completed? Yes or no? Abir, are you facing some internet issues? Okay, students, tell me one thing. Apart from all of the chapters that we have completed up until now, is there any other chapters some of you have in your exam other than these? Yes or no?
Ma'am, I have unit test on this one.
You-
Ma'am, I have excretory products and their elimination, body fluids and circulation.
Body fluids and yeah, I was thinking about starting body fluids and circulation. Okay, it's a very nice chapter. It is my favorite chapter. Okay, so all of you will have to listen to it with a lot of interest. Okay, I'm giving you a heads up. Abir, are you facing internet issues today?
Yes,
I
ma'am.
can see that. Okay, what is the last thing that you people have written in your notebooks? The differences is completed or is it left? Completed or left? Tell me. Don't wait for the others to answer. Someone tell me. How would I know what you have written?
Ma'am, we finished writing the difference
The differences were over, right?
between
For everyone?
C4 and C3.
Okay. All right. Now you have to start from photo respiration. Okay, students. Now you can switch off your videos. Write down comfortably.
Done, ma'am.
All of you are done, students?
Yes, ma'am.
What about the others? Are you also done?
Yes, ma'am.
Oh.
Ma'am, can you please scroll down?
All of you are done, students?
No, ma'am. Just a minute.
Okay, okay.
Done, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Just a minute, ma'am. Done, ma'am. Done, ma'am.
Done, ma'am.
All of you are done, students?
Done, ma'am.
All of you are done? Okay, so today we're going to start a new chapter. Do you people have any doubts in photosynthesis? Yes or no?
No, ma'am.
No.
All right, so we're going to start with a new chapter. Don't even have the heading. Let's write that also. Also, everyone, now you can switch back your videos on. So our chapter is body fluids and circulation, right? This is the name of our chapter. So what we're going to do. Now, the first thing we know is that there is blood in our body, which is almost everywhere, isn't it? Is there any place in our body which does not have blood? No. And the reason why it's everywhere, because the blood is the fluid connective tissue. I hope that you people remember it from class 10. Fluid connective tissue. Now let us see what are the what is the composition of the blood, meaning what the blood is made up of. Now the blood has two components, two major components. The first one is the plasma. The second one are the elements, the formed elements. Plasma comprises 55% of the blood, and the formed elements, they comprise 45% of the blood volume. Now, plasma, we'll discuss it, but when we move forward under the label of formed elements, so we have the blood platelets, right? And we have the blood cells. These are two different things. Remember that the platelets are not cells. Okay, we'll see what it is and what it is not. There are so many good things to this chapter, students. You people are going to enjoy a lot. This is a very nice chapter. All right. So talking about the blood cells. So we have erythrocytes, that is the RBC, that is the red blood cell. And then we have the leukocytes, that is the WBC, white blood cells. Okay. Now, each of these components of the blood, they have their own very particular, very specific, you can say, function to perform. Now, the RBCs, they are not further divided into any types, but these WBCs, they have a great variety. Like our body has a great variety of WBCs in it. Now, WBC, they are of two types, granulocytes and agranulocytes. This is with respect to their appearance. Granular appearance, you understand? Rough appearance, sort of. And agranular, meaning it's not a granular appearance. So now under the granulocytes, you have the neutrophils, there is the basophils and eosinophils. Okay, I say it eosinophils because, while I was in class 11, to remember the spellings, I used to pronounce the word just like the spelling that they had. So it's not eosinophil, it's eosinophils. But I have this habit of calling it as eosinophils so that I can remember the spelling. So you people can do that too. Now talking about the agranulocytes. So they are further of two types, monocytes and lymphocytes. And ultimately we have the most important cells, that are the B lymphocytes and the T lymphocytes. If these two cells are not there in our body, we'll die. They are the soldiers of our body. In general, WBC lot is the soldier of our body. This composition of blood is clear to you all. Obviously, don't expect that it'll get inside your brain or get imprinted at once. No, you'll have to take a while to see it, to see what are the divisions. But this chapter will swiftly go into your mind. Trust me. Now, The first thing that we are going to talk about is the coagulation of blood, that is blood clotting. See, okay, and one thing I wanted to tell you people is that this chapter is not in a very organized like sequence. Every topic is there, but it's not how it's given in the book. But all the topics I'm going to discuss in depth actually. Now see, talking about coagulation of blood. Coagulation means clotting. Now we know whenever we get a cut, whenever we get a serious injury also, and even if it is a scratch, if blood comes out, right? So in that case, for example, if we have gotten a deep cut, now the blood does not keep on flowing forever. Imagine you got a little paper cut and the blood keep on flowing and it's not stopping. Will die, right? So as a defense mechanism, or you can say as a protective mechanism, our body has this very unique property of coagulation of blood. Okay, so whenever we get a cut, we get a wound, in that like for the blood flows for a little while, but then it stops, because obviously any kind of reaction in the body, it needs time to take place, right? So blood clotting happens, and you might have seen that like wherever you get a cut or a scab or a injury, after the blood clotting has been done, there's a very thick layer formed on it, thick layer of tight cells that are formed on it, right? Which we are very tempted to scratch it off, right? You might, I have it here also. I'm not scratching it off, but it's like we are so tempted to scratch it off, and that is the, you can say, the work, the hard work of our platelets. That blood platelets I told you about, it is one of the important components of the blood, and this coagulation of blood or this blood clotting mechanism is the most important function performed by the platelets. Okay, they are the ones because of which our blood clot happens. Now students, I have a question. If the blood will clot, then why does the blood not clot in our body? The blood has platelets, right? In our body also it has platelets. So why does it not clot inside our body? Why do you, well, what do you think about it? Tell me. See, in our body, there is the presence of anticoagulant proteins. It does not let the blood clot inside our body. But as soon as if you take out a drop of blood from your body and you put it on anywhere, it would clot. It will not be a liquid form, rather it would become a sticky gel-like structure. Now, the process through which the blood changes from a liquid and becomes thicker like a gel. This happens in the case of injuries. Now, this is a mechanism to prevent the excessive loss of blood from the body. Now, a dark reddish brown scum is formed at the site of the cut or the injury. That thing that we are so tempted to scratch it off, that is a, you can say it is a clot, or you can call it as a coagulum. Now, this coagulum that we have, it is not just something, you know, like it is easy for us because we scratch it off, but we don't know what goes behind it, right? That clot or that coagulum, it is made up of a network of fine threads called as fibrils, meaning these are the proteins in which the dead sort of like, imagine those proteinaceous structures, they form a net like this, so that whatever is the damaged area or damaged cell or the damaged blood cell, it gets caught in that network like fibril. And for the blood clotting, the enzyme thrombin is present in the plasma of the blood. Remember, I said that plasma is one of the components of the blood that comprises 55%. This thrombin is present in the plasma of the blood. Okay. Now, what happens during the blood coagulation? Let us see that. Now, whenever there is any injury, any cut, any wound, that stimulates our blood platelets to release certain, you can say, proteins called as thromboplastins. Okay, thromboplastins are coagulation promoting substances. So as soon as the cut or the wound happens, our blood platelets in our body, they get activated and they start releasing thromboplastin. Now this thromboplastin, what it does is that it starts activating the whole entire blood clotting mechanism. Okay, it sends the message to the body that, okay, now we have to clot the blood. Now the tissues, like first is that the platelets release the thromboplastins. Now the tissues in that area, the area that got cut or wounded, the tissues of that area, they also release the tissue thromboplastins. So two kinds of thromboplastins are released. One is released by the blood platelets, the other is released by the tissue. Okay, this much is clear. Now the next, this thromboplastin, it helps in the formation of the enzyme complex called as thrombokinase. Okay, this thromboplastin forms thrombokinase. This is like, imagine it like, have you people seen that? I think it's called as domino, that you place the small blocks and if you push one, all of it will fall, right? That is a domino. Same way, this whole entire mechanism of blood coagulation is just like a domino. It's like a cascade mechanism. One after the other, one after the other, it is linked to each other. So now the thromboplastin, they help in the formation of the enzyme complex called as thrombokinase. Now this complex is formed by a series of linked enzymatic reaction, and this is a cascade process. Cascade means domino-like. This is just for your understanding. Don't write domino in your exam and don't write dominoes in your exam, more importantly. Now, an inactive protein called as prothrombin is present in our plasma. Now, in the presence of thrombokinase and calcium ions are also there in our body. So, in the presence of thrombokinase and calcium ions, the prothrombin prothrombin is inactive, right? So, it will get activated in the presence of calcium dipositive ion and the thrombokinase. So, now it has become an active thrombin. Further, do you see how one enzyme is being created and that is helping in the synthesis of another? Same way, one will be created helping in the synthesis of another. So now this thrombin that is created, now this thrombin will be responsible for converting the fibrinogen in the plasma to fibrin. So again, fibrinogen is inactive and in the presence of thrombin, now it will get converted into fibrin that is active. Like these are all the proteins that are present already in the plasma, but in the inactive form, right? If it will be active, then the clotting of the blood will be done inside our body only. So that is why it is inactive. It only gets activated whenever there is an injury. Now this fibrin only, it forms a mesh-like structure or a network-like structure, and it traps the dead and broken elements of that area wherever we've gotten the cut to form the blood clot or the coagule. And this clot seals the wound and stops the bleeding. And we know that the calcium ions, they have a great role because they are acting like, you people remember from biomolecules, coenzymes. Remember coenzymes? So here the calcium would act like a metal ion or ion that acts like a coenzyme. Okay, it is increasing or enhancing the reaction. Is it clear, students? The whole entire mechanism? It's okay if you did not learn it right here and now, but the mechanism is clear to you, how it is happening, how one thing is being formed, and then that one thing is forming another thing, and that one thing is forming another thing. So that is like a whole series of reaction. Is it clear, students?
Yes, ma'am.
All right, very good. Now you people can note it down. Can you see this clearly? Or shall I zoom?
Ma'am, zoom. Can you zoom?
Okay. All right, now you people can note it down.
Done, ma'am.
Done, students?
No, ma'am.
Okay.